Hi, I'm Srivarshini Ganesan and my research focuses on understanding the aggregation of proteins in Alzheimer's disease. Alzheimer's disease is a neurodegenerative disorder in which a particular protein gets aggregated and deposited between the neurons, preventing the neurons from functioning properly and leading to neuronal death and shrinking of the brain. Alzheimer's disease leads to memory loss, cognitive impairment, trouble with language, speaking properly, doing familiar tasks, frequent mood swings, and behavioral changes that eventually lead to death. Alzheimer's disease is very prevalent, affecting more than 40 million people globally, and you'd be surprised to know that there are no treatments yet that can stop or reverse the disease progression. This is because we still haven't clearly understood how the disease occurs or progresses. Now, to gain a deeper understanding of how the disease occurs and progresses, we have two different techniques that we can use. One, you can use an experiment by extracting the diseased proteins from the diseased tissues and then visualizing under electron microscopy or using techniques like X-ray crystallography or NMR. You can understand how the proteins are getting aggregated. Or you could use a computer to simulate this and understand. That sounds interesting, right? And our lab does exactly this. We use computer simulations to model protein aggregation. In computer simulation, all the atoms in the system are assumed to be point masses, and the bonds and angles between them are assumed to be simple harmonic oscillators. And then using Newton's laws, we can calculate where a particular atom would be at the next time step. You know where it is right now, all the forces that are acting on it, then you can easily find out where it would be at the next time point. So like this, for all the atoms in the system throughout the time frame, we'll find out where it is located. And this is given to you as a simulation trajectory data, which I'll be showing you now. And this actually comes up as a movie. You'll be exactly able to see how the initial structure goes on to become the ordered fibril structure. I couldn't show you the movie here, so this is a snapshot at the beginning, at the intermediate state, and at the final step. So with this visualization, you'd be able to get insights about how this aggregation occurs. Furthermore, we do a series of analysis to get deeper insights and other more parameters that actually drive aggregation. And with these analysis, we'd be able to understand the pathway of aggregation and also design drugs that can go bind to these proteins and prevent their aggregation and find out drugs that can actually cure Alzheimer's disease. Thank you.